if we consider the present scenario of the development in the chemical sciences. As the word sustainable itself is very important. Sustainable development is one of the most frequently used term over the world politics, we can say, because it is the very important and current area in the chemical sciences. Because what before going to the actual content of the seminar or starting the seminar, we must know what is sustainability. It is the development that meets the need of present generation by considering the future of incoming generation without disturbing the ecological systems. So this is very important if we consider the research in the chemical sciences. Now, for today's wonderful webinar, we have got two eminent resource personalities. One is from Italy, Professor Antonio Massa, Department of Chemistry and Biology, University of Palermo. And another one eminent resource persons are Dr. Bapurao Shingte from Department of Chemistry, Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad. Now, I first of all welcome all of you, and I request Principal Dr. Chandrakant Javade sir, Rasika Mahavidyalaya Devni, to give their introductory talk to this international webinar.
Thank you, sir. Good afternoon, everyone. The president of this function, Honorable Sriman Mr. Govindraoji Bhopnikar Sahib, Jan Seva Sevavayu Pratishthan Bhopni, and Honorable Chairman of Mr. Santoshji Jangam sir, KTSP Mandal Khopoli, Honorable Vice Chairman Mr. Rajeshji Abhani sir, KMC College Khopoli, and Honorable Guest today functions Mr. Gajananji Bhopnikar sir, Secretary of Jan Seva Sevabhavi Pratishthan Bhopni, Honorable Mr. Kishore Patil sir, Secretary of KTSP Mandal Khopoli. Inauguration movement of international online webinar in the hand of innovator, Honorable Professor Dr. L.M. Wagmare sir, Dean of Science and Technology, Faculty Science, Swami Ramanand Tirth Marathwada University, Nanded, Chairpersons of International Online Webinars, Honorable Professor Dr. B.S. Downe sir, Director of School of Chemical Science and Chairman of BOS, Swami Ramanand Tirth Marathwada University, Nanded, Honorable Principal Dr. Mahesh G. Khanvilkar sir, KMC College Khopoli, and Organizing Committee Member Professor Dr. Vijay Kumar More, Mr. Ankush Bhaskar, Kailaswasi Rasika Mahavidyalaya Devni, Latur, Professor Dr. Bharat Panchagalle, KMC College Khopoli, and other IQAC member Professor Ankush Bhusawale, Dr. Deepak Gaikwad, Mr. Amol Nagargoje, and specially international online webinar invited speaker Honorable Mr. Professor Antonio Massa, Sir, Department of Chemistry and Biology, University of Salerno, Italy, and second invite speaker Honorable Professor Bapurao Shingte Sir, Department of Chemistry, Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad, and all eminent participants of this international webinar of professors, teachers, students, researchers, and professional industries body of members. It is my great pleasure to welcome you in this memorable moment of this online international webinar occasion. I am very grateful and thankful in our educational society to give me such a special opportunity to organizing the permission of invite international webinar. Today, I am going to talk few words about of my college. Devni town is popularly known for Devni Vadu bulls, famous throughout the country as well as the world. Our college located at Devni, district Latur in Maharashtra state, India. The college has been established mainly for Marathi and Kannada speaking people in the border disputed villages in the Maharashtra and Karnataka state of India. This college is young and established in the year of 2008. The college is recognized by government of Maharashtra and affiliated to Swami Ramanand Tirth Marathwada University, Nanded. The college has independent, spacious, and attractive building. All science department have well equipped laboratories. Our college has been included under Government of India Ministry of HRD under the UGC 2F and 12B. Our college is imparting the higher education to economically and socially backward class students of rural and remote area. It offers PG and UG program leading. Our college is engaged in the sustainable overall development of the student under the guidance of Honorable Sriman Govindraoji Bhopnikar Sahib. He is dimensionally persons and its own master plan for the future developing. He promotes of the faculty members and monitors various activities for imparting quality education all over rounder development of the student for achieving their vision and mission. Now, I am going to talk about international webinar on the innovative research in chemical science for a sustainable development. Innovation that use the existing knowledge and innovations to create a new technology can be facilitated. The most successful facilitation occurs through the creation of an idea that increases many companies to grow innovation and important part tend to focus on technique to generate new ideas for innovative product. The chemical process industries and chemical professions are always viewed by the general public as being very innovative fields. The general public does not understand the key role of chemical science. Innovations play in the providing food, water, energy, good to the consumer. To remain viable and competitive, the chemical process industries must innovate every day beyond just standard research and development activities. The chemical science have new sustainable development to improve the efficiency with which natural resources are used to meet human need for chemical products and services. Sustainable development of chemical science encompasses the design, manufacturing, use of efficient, effective, safe, compact, flexible, and more environmentally benign and conducive to the rapid commercialization of a new product, new challenges for a chemical science. As a society, we need to start telling people that it is not only these fancy achievements that make a person successful and useful to society. Lastly, I hope that this platform for a meaningful deliberation will be highly useful to the student, budding scientists, industries, businessmen, teachers, professors, researchers, as well as a good public sector service. I hope that success of the international webinar on innovative research in chemical science for sustainable development. Thank you. Jai Hind, Jai Bharat.
Thank you, Dr. Chandrakant Javade, sir, Principal of Kailaswasi Rasika Mahavidyalaya Devni. In your introductory speech, you have clearly elaborated the purpose of today's international webinar. Now I request the principal of our Khopoli Municipal Council College, Khopoli, Dr. Mahesh Khanwilkar, sir, to give their introductory speech for this international webinar.
Thank you, sir. Good afternoon. Good afternoon, everyone. Respected dignitaries, invited speakers, teachers, and my dear students. Welcome to the today's international online webinar jointly organized by Kailaswasi. Good afternoon, everyone. Respected dignitaries, invited speakers, teachers, and my dear students. Welcome to the today's international online webinar jointly organized by Kailaswasi, Rasika Mahavidyalaya Devni, and AMC College of Khopoli. To me, it's an immense pleasure. To me, it's an immense pleasure to get an opportunity to conduct this international webinar in our college, which was established in the year 1979. under the esteemed guidance of Honorable State Minister Mr. B.L. Patil sir. Our college is well reputed old college in Raigad district, which is referred as aided college from beginning itself. Chemistry department has been the largest department here, having specialization in UG level to PhD level. And EST fees has sanctioned 70 lakhs rupees for research purpose for science faculty. Under that well-equipped research laboratory is established. I am very much grateful to all the members of KTSP Mandal for their continuous encouraging attitude. Thanking you all.
Thank you, sir. Thank you, sir, for the kind introductory speech for the today's webinar. Now, for our today's international webinar, we have inaugurator Dr. L.M. Wagmare, sir, Dean of Science and Technology, Swami Ramanand Tirth Marathwada University, Nanded. Now, I request Mr. Ankush Bhaskar, sir, for to give the kind introduction of Professor Dr. L.M. Wagmare, sir.
Hello, good afternoon, everyone. Here, it's my pleasure to introduce Dr. L.M. Wagmare, sir, the inaugurator of today's international webinar, which is organized on innovative research in chemical science for sustainable development, which is organized by the Department of Chemistry and IQAC of Rasika Mahavidyalaya Devni in the collaboration with the Department of Chemistry and IQAC of KMC College Khopoli. Dr. L.M. Wagmare sir, the Dean of Faculty of Science and Technology of Swami Ramanand Tirth Marathwada University, Nanded. He has completed PhD in 2001 from IIT Roorkee. In his service, he has 34-year teaching experience. He has 11 students have completed PhD under his guidance, and 8 students are doing research work in progress. He has, to his credit, 1 book, 3 book chapter, publications of 55 plus paper in international journals, and 60 plus papers in national and international conferences. He is a recipient of K.S. Krishnan Award by Institute of Electronics and Telecommunication Engineering in 2004 for the best system-oriented research paper. Before joining as a Dean of Science of Faculty of Science and Technology of this university, he worked as a director of Sri Guru Gobind Singh Institute of Engineering and Technology, Nanded, and a Dean of Faculty of Engineering and Technology at SRT University, Nanded. He also worked in various capacities such as Head of the Department, Dean of R&D, Dean of Academics. He visited the University of Technology, Petronas, UTP, Malaysia in 2013, City College, University of New York, Oakland University, San Jose State University, California, for signing a MOU and discussion with the academia for collaborative research, student and faculty exchange program. He visited Australia, China, Malaysia, Singapore, Dubai for presenting papers and attending workshops and discussion with academy. He worked on various committees such as Board of Studies, Academic Council, Management Council of Executive Council or Executive Council of Board of Management, Senate, etc. of various universities. Now, from 29 July 2019, he is a Dean of Faculty of Science and Technology of this Swami Ramanand Tirth Marathwada University. So, as we get such a great personality as an inaugurator of today's international webinar, on behalf of the Department of Chemistry and IQAC of Swami Rasika Mahavidyalaya Devni and KMC College Khopoli, I most welcome to all the guests, all resource person and dear participants. Thank you very much.
Thank you, sir. Now I request Professor L.M. Wagmare, sir, to give their inaugural speech for this international webinar.
Good afternoon, one and all here. First of all, I extend my sincere thanks for introducing me in a very nice way. I also express my sincere thanks for giving me an opportunity to be the part of this event. All the honorable members, those who are attending the webinar, all the honorable members of both the colleges, members from the management, principal of both the colleges, members of IQAC cell, organizing team, and honorable international experts. First of all, I, on behalf of university, also express my sincere thanks to all the experts, those who are going to share their expertise with the audience, especially the experts from the international universities, because they have to join early in the morning this event. And I'm sure that their expertise will help us to give a thought in a different way. First of all, I congratulate the coordinators of both the institute, both the colleges, for organizing the event on a theme which is the need of time. If you look at the theme of this particular webinar, international webinar, 3 keywords are altogether having their different importance. Sustainable development has to be of importance, is of importance at all the levels. Any kind of development without sustainable planning will not help. So sustainable development is the keyword, buzzword that is used in this one, and that is combined with the innovative research and chemical sciences. Chemical science, we know the importance of chemical sciences in the life of every individual, life of common person. So we are giving a thought to the research to be carried out, to be done, which is very much having the relevance to the social importance, relevance of the social importance. And particularly innovative research. Innovative research will lead to develop a product, to develop a concept which is of a great need to the society. Innovative research will lead to the patent, will lead to the filing a patent or granting a patent and giving a ownership to a particular concept. I personally believe this kind of workshop help us to understand the scope and status of the particular concept at the national and international level, which will help us to define the research problems. See, basically, if we want to work on some concept, we should know on what we should work. So this kind of forums, this kind of discussions will always help us to identify the research problems, to identify the status of the remedies for similar kind of problems at national and international level. The another thing which is very important that should come out of this any kind of international webinar is the networking of experts in a specific domain across the globe. This webinar basically help us to bring the different experts on a common platform, and we should not conclude our relationship, we should not stop our communication with this webinar itself. It is a need of time that we have to have the networking across the globe with the experts for developing the different concepts, for coming out a research of a great value. Another advantage of having similar kind of workshop is collaborative research. If we identify the experts from the similar domain, we can have the collaborative research, for example, from collaborative research between the different universities at the national level or different colleges at the national international level. So that will help us to broaden our vision. That will help us to take the give the visibility to the our colleges across the globe, university across the globe. So I believe that by the end of the day, we'll have the remarkable development, remarkable conclusions on this front. With these few words, once again, I welcome all the experts at national, international level, and express my sincere thanks for giving me an opportunity to be part of this event. I also declare that the webinar on innovative research in chemical sciences for sustainable development is inaugurated, is open for the discussion. Thank you very much. Thank you, sir. Please.
Thank you, Professor Wagmare sir, for giving the kind inaugural speech for this international webinar. Now we have with us Professor Dr. B.S. Davne, Director of School of Chemical Science and BOS Chairman. So first of all, I request Mr. Ankush Bhusawale sir to introduce the chairperson of today's international webinar, Professor Dr. B.S. Davne sir. I request Mr. Ankush Bhusawale sir to introduce our chairman of today's webinar.
Thank you, sir. Good afternoon to all the dignitaries and participants of this international webinar. It's my great honor and pleasure to introduce today's webinar chairperson, Honorable Professor Dr. Bhaskar Sadashiv Davne, Director of School of Chemical Science, Swami Ramanand Tirth, Marathwada University, Nanded. His educational qualification is MSc, PhD, and his specialization is in the area of organic chemistry and medicinal chemistry. His major thrust area of research is synthetic organic chemistry and medicinal chemistry. He has more than 30 years of research and teaching experience. Until now, he has published more than 90 research papers in international peer-reviewed journals. Many of his research papers were cited by other research scholars. One of his research paper has got Best Research Paper Award in 2014. He is research guide, and under his guidance, many of the students has been awarded with PhD and MPhil degrees. He is also a BOS member in the university. It is really impossible to mention each single accomplishment achieved by him. His achievement is recognized by the Maharashtra state by honoring him a Best Teacher Award at state level and postdoctoral research award from University Grant Commission. He also has actively engaged in various social activities. He has accepted our invitation as a speaker on this occasion of international webinar. Thank you so much, sir, for joining with us with your own presence. Thank you for being with us. I request Honorable Dr. Davne sir to please share his valuable thoughts and experience with our webinar participants. Please welcome Honorable Professor Dr. Bhaskar Davne sir. Thank you, sir.
I request Professor Davne sir to share their chairperson's remark for this webinar.
Davne sir, we can't hear you.
Yes, sir.
Okay, now we can hear. Okay.
Okay, sir. Good afternoon, everybody. Inaugurator of today's wonderful international online webinar on innovative research in chemical science for sustainable development. Honorable Professor Dr. L.M. Wagmare Sarji, Dean of Science and Technology. Today's invited speaker, eminent scientist, Professor Antonio Massa, Department of Chemistry, Biology, University of Salerno, Italy. Second eminent scientist, well-known personality of our area, Dr. Bapurao Singates, Department of Chemistry, Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. Chief patron of today's international online webinar, Honorable Govindraoji Bhopanikar, President of Jan Seva Seva Bhavi Pratishthan. Dr. Gajanan Raoji Bhopanikar, Secretary. Honorable Santoshji Jangam, Chairman KTSP Mandal Khopoli. Honorable Kishorji Patil, Secretary. Honorable Rajeshi Abhani, Vice Chairman. Organizing Committee, Dr. Vijay Kumar More, Assistant Professor, Kai Rasika Mahavidyalaya Devni. Dr. Chandrakant Zavle, Principal, Kai Rasika Mahavidyalaya Devni. Dr. Mahesh Khanvilkar, Principal, KMC College, Khopoli. Dr. Sharadji Panchagale, KMC College, Khopoli. All other organizing committee members and all participants joined in international online webinar. Firstly, it is my great pleasure to welcome all eminent scientists and participants joined in this webinar. Really, from my bottom of heart, I appreciate to organizing committee to select very important subject in chemistry, that is sustainable development. Just Professor Dr. L.M. Wagmare sir and Principal Chandrakantji Zavle sir express his thoughts about this innovative research. So we know that sustainable development goals are directly related to the necessity reducing the impact of chemicals on the component of environment, eliminating dumping and minimizing release of hazardous chemicals and materials, having the proportion of unreacted wastewater, and sustainably increasing recycling and safe reuse globally and project our ecosystem. We know that due to the COVID-19 pandemic situation, tremendous use of PPE kits, so there is a great problem of decomposition and recycling safely of PPE kits. So I hope this international webinar is very useful to all participants for the sustainable development in chemical science. Lastly, I express my thanks to organizing committee to give me opportunity as a chairperson for this international webinar. So I last conclude my speech. Thanking you. Thank you.
Thank you, Professor Daune sir, for the kind remark for this international webinar. Now, for this today's international webinar, we have with us our Honorable Secretary of Khalapur Taluka Shikshan Prasarak Mandal Khopoli. So I request Honorable Mr. Kishore Bhai Patil to express their thoughts for this international webinar.
त्यांचा आवाज येत नाही आता काय तर नका सांगायचं आता. पाटील सर, we can't hear you. आता जाऊ देत सर, काय आता सांगितलं ना.
Hello, Patil sir, your voice is not there.
Oh.
I think no, he is not audible for the audience. We'll move further, and when he join, we will take their remarks for this webinar. Now, I request Dr. Vijay Kumar More sir to introduce our today's first resource person, Professor Antonio Massa. I request Dr. Vijay Kumar More sir to introduce our first resource person for today's international webinar.
Good afternoon, all of you.
It is a great honor for me to introduce and welcome the today's webinar resource person, Professor Antonio Massa, Department of Chemistry and Biology, University of Salerno, Italy. He has completed master degree from University of Salerno. After that, he worked in company for 3 years at Montefibre in Naples, Italy, as a young researcher. Then he has joined for PhD at the University of Salerno under the supervision of Professor Skriti as a diastereo on diastereo and enantioselective selective oxidation and carbon-carbon bond formation. During his PhD, he was visiting students at University of Glasgow, Scotland. Also, he did postdoc from University of Salerno, Italy. Then he joined as an assistant professor at Department of Chemistry of University of Salerno. He was awarded the Fulbright Research Scholarship from Scripps Research Institute, San Diego, California, as visiting investigator in the laboratory of Professor Carlo Barbas, and he is a visiting professor at the Institute of Organic Chemistry, University of Linz, Austria, and Institute of Organic Chemistry, Munich, Germany. He is in the editor board of many international reported journals like Molecules, edited by Springer's Asymmetric Catalysis, Journal of Chemistry, Current Catalysis, Current Organic Chemistry, edited by Bentham, etc. Also, he is a reviewer for many journals like Tetrahedron Letter, American Chemical Society, Chemical Review, Organic Letters, Journal of Chemistry, Royal Society of Chemistry, Chemical Science, Advanced Synthesis and Catalysis. Since 2015, he is working as a professor at the University of Salerno, Italy. He is a supervisor many students for master and PhD student thesis. One of these, I was very lucky. I am his students. He is a published more than 19 articles on international journals. Also published 2 patents and several books chapters. His research interests are focused on the development of new methodologies, asymmetric catalysis, organocatalysis, sustainable one-pot reaction, and the synthesis of bioactive compounds. He is also coordinator of research project in collaboration with national and multinational companies. So we get such a great personality on as a resource person of our webinar. So on behalf of our chemistry departments and all participants, of most welcome, sir. Thank you, sir.
Thank you, Dr. Vijay Kumar More, sir, for your kind introduction of today's resource person. So before actual session,
सर आवाज येतोय माझा सर?
Yes, sir. Yes, sir. Before actual session of our today's first resource person, we have with us Honorable Secretary of our KTSP Mandal Khopoli. So I request Mr. Kishore Bhai Patil Sahib to express their thoughts for this international webinar.
Thank you, sir. Good afternoon to all. Nowadays, the whole world is suffering from COVID-19 pandemic. Many countries announced lockdown to overcome pandemic situation. Our country, India, is not exception for it. Since March 2020, we are facing lockdown. Intellectual people have habit to interact with each other. The exchange of thoughts, the interaction is through conferences, seminars, etc. Many of the faculty members of KMC College are actively involved in research, and they present their research finding in reputed journals national, international conferences, symposium. Many of our teaching staff got prizes in conferences for best oral presentation, best poster presentation. In the present situation, it is impossible to organize or to attend physical gathering for interaction. Smart people of the world gave solution on it in form of web meetings, webinars. KMC College organized many national webinars in past as office bearer of parent organization KTSP Mandal. I feel proud of our KMC College, Khopoli, for organizing international webinar. Always encourage in organizing such events. As per my knowledge, in a chemical reaction, weak bonds break and new bonds come in picture. The webinar is like chemical reaction only. I hope through this international chemistry webinar, there will be subtraction of old outdated thinking and audition of new ideas. I also feel there will formation of new bonds between people. On this occasion, on behalf of KTSP Mandal, I virtually welcome both resource person, Professor Masa of University and Celerano, Italy, and Dr. Singate sir of Dr. Babasaheb Ambedkar University, Aurangabad. With this, I conclude my remark. Thank you. Thank you very much, sir.
Thank you, Mr. Kishore Patil, sir, for their kind, speech for this international webinar. And also thank you for their consistent efforts in developing our college and throughout efforts. Now, without taking much time, I request our resource person, first resource person, Professor Antonio Massa from Department of Chemistry and Biology, University of Salerno, Italy, to deliver his vital lecture for today's international webinar. Professor Antonio Massa.
Okay. Thank you for your kind invitation, and also I want to thank Vijay. He's not only was very good student, a PhD student. We did very nice research in the past, but he is also very good friend of mine actually. And let me share my presentation. I will try to share the. Okay. Please tell me if everything is fine. Okay. Do you see presentation mode?
Yes, sir.
Okay. So do you hear me clearly? Okay, so I can start.
Yes.
Okay, thank you. The name of the presentation is Principles and Application in Asymmetric Organocatalysis. Actually, organocatalysis is considered a field of green chemistry, and we can see that later on in the presentation. So this fit very well in your international webinar, and in the first part of this presentation, I'm going to talk about the principles, the general applications of asymmetric organocatalysis. In the second part of the presentation, I will show you some recent research results from my group in this field. Okay, let me go on. Let me show you a little bit of history in this field. These are very, how can I say, very dated articles. As you can see, they can be dated early at the beginning of the last century, in 1912. They were always also studying organic reactions catalyzed by small organic molecules, as you can see here. Let me, sorry, I need the pointer. Okay. Do you see the pointer? Okay. So in this first reaction, this is a little bit of history. You can see hydrogenation of benzaldehyde promoted by a cinchona-alkaloid derivative. So this can be considered actually an organocatalyzed reaction, even though the enantioselectivity was very low, but we are talking about beginning of the last more than 100 years ago. Later on, a few other papers appeared in this field as this reaction between methanol and ketone, as you can see here, formation of this ester, also in this case catalyzed by derivative or cinchona-alkaloids and with good enantioselectivity. Also in the 70s, another important reaction appeared in the literature, in this case catalyzed by L-proline, an amino acid. This is intramolecular aldol reaction, leading to this very important building block in very good enantiomeric excesses. These are the only 3 papers, important papers, but they were also they were also almost forgotten until 2000. 2000 is the starting date for organocatalysis. And in particular, almost at the same time, 2 very important articles appeared in literature. This first one, authored by List, Lerner, and Barbas III, as you can see, this is a famous cross-aldol reaction, very simple reaction between acetone and aldehyde. In this case, the catalyst was L-proline, and the product is an aldol product, was obtained in a very high enantio purity, up to 96% E, especially when the aldehyde is aliphatic aldehyde. Almost at the same time, another very important author, Macmillan, reported this very important reaction. This is a Diels-Alder reaction catalyzed by this organic compound known as imidazolidinone. This article, reported by Macmillan, is the first paper mentioning the term organocatalysis. So this is very actually important, also because in this paper, they rationalized the mechanism of this reaction. And we will see the mechanism of these reactions later on during this presentation. After 2000, a huge number of papers appeared in literature employing small organic molecules as chiral catalysts in asymmetric reactions. Depending on the interactions between the catalyst and the substrate, they can be listed as several subfields, as you can see here, depending on the activation mode. In particular, enamine catalysis, iminium ion catalysis, hydrogen bond catalysis, asymmetric phase transfer catalysis, also asymmetric counteranion catalysis, asymmetric photocatalysis, Lewis basis catalysis, bronze acid catalysis, carbene catalysis. All these names represent a mood how the catalyst can interact with the substrate, promoting the reaction in a symmetrically way. And this is a short list with some structures of organocatalysis. Actually, one of the most important organocatalysts belong to these 2 classes. Covalent catalysts, in which you can see L-proline and some derivatives of proline, like prolinol protected with the 3-methylsilyl group, or non-covalent catalysis. You can see here some important derivatives. In particular, the first 2 are based on cinchona-alkaloids compounds, derivatized, in this case with thiourea, and in this case, this is a chiral ammonium salt. You can see also another important organocatalyst, and this is a chiral phosphoric acid. In this second part of the list, the interactions between these compounds and the substrate are non-covalent, so can be ionic interactions or hydrogen bond by staking, dipolar, and so on. And this is another list of covalent organocatalysts You can see also proline reported for the first one by List and Barbas, and also the prolinol reported from Ayash and Jorgensen, and these are the Macmillan imidazolidinol. Now, let's go a little bit in details about the mechanism. So, enamine catalysis is particularly effective for cross-aldol reaction, as you can see in this slide. And this is a very effective reaction between acetone and an aliphatic aldehyde. So, since the acetone is used in excess, and so cross-aldol reaction is more favored than self-aldol condensation. The reaction is promoted actually by proline, and turnover is not so high because, as you can see here, proline is used at 30%. But anyway, proline is very cheap actually. This is the mechanism. The first step is the interaction, the reaction between the proline and the acetone is condensation. With first you will see the formation of the aminium ion, and then this is tautomer equilibrium, formation of enamine. Enamine is the effective nucleophile in this reaction, is nucleophilic species. But however, the carboxylic acid group is very important because through hydrogen bond, it will form hydrogen bond with the aldehyde. So this is very tight transition state in which nucleophile and electrophile are very close and they can react in a very selective way. So this is a chair-like transition state. So the R group of the aldehyde is supposed to be in pseudo-equatorial position, therefore it's possible to obtain the aldehyde product in a very stereoselective manner. Anyway, after this reaction, the product, the intermediate, is an iminium ion. However, since in the first part of the reaction, water is a byproduct, in the second part of the mechanism, water is very important to allow the hydrolysis of this iminium ion to free the aldehyde product, and most importantly, to free the proline, which can start a new catalytic cycle. And this is the same mechanism, actually, a little bit more details in the first part about mechanism, but we can skip this mechanism actually. And what is important is the selective attack in the transition state of the enamine to the RE phase of the aldehyde. The other transition state in which the R group is in a pseudo-axial position is net favored, is a high energy transition state. It's very important that all these reactions have been recently reviewed in the book, in this very important book, Green Catalysis, edited by Anastas, which is one of the really expert in sustainable chemistry, as you know. What is this? Anyway, this is another evolution of this cross-aldol reaction. You can see the same reaction, but in this case, the ketone is a symmetrical ketone, is a methyl ketone, can be 2-butanol. In this case, the reaction is also highly regioselective because the reaction is under thermodynamic control, so it is favored the enamine, the more substituted enamine. The reaction is also very high diastereoselective, leading to the anti-diastereomer in very high ratio, as you can see here, and very high enantiomeric purity, 99%. The R1 group is methyl group actually. But what about the cross-aldol reaction between two aldehydes? This reaction has been developed by, this is also my friend Ayashi, Yujiro Ayashi from Japan, is also, he also proposed this type of organocatalysts. And I have to say also that Carlos Barbas III, I've been at the Scripps Research Institute in California for a postdoc in 2008 as a Fulbrighter with a Fulbrighter fellowship, and Carlos was a really very nice person. Because Carlos unfortunately passed away in 2013 only at the age of 49. So this for me was very big. Anyway, let me go on. And also this reaction was reported by, because this is a very important reaction, very challenging reaction, because you have to imagine a cross-aldol reaction between two aldehydes. And so this reaction is also very selective, even if the yields are not so high, but you can always isolate the aldol product after reduction of the aldehyde group, in situ reduction of the aldehyde group, in very high enantioselectivity. selectivity. So let's move a little bit to analyze another organocatalyzed reaction, in particular Manic reaction. Manic reaction is a very important reaction because of the possibility of introduction of nitrogen atoms in a very complex scaffold, and the nitrogen is always present in the bioactive compounds. There are two ways to perform asymmetric Manic reaction. Between pronucleophile, as you can see here, the acetone, and the preformed amine, as you can see here. And also this reaction is catalyzed proline. Do you hear me?
Yes,
okay.
sir, we can hear.
Okay, I can go on. Also, this reaction is catalyzed by proline interacting with acetone, formation of enamine, giving the final Manic base in very high yields and very high enantioselectivity. Another very clever way to perform this reaction, also reported by my friend Carlos Barbas, is one-pot reaction. Instead of the synthesis of the amine, what happens? This is one-pot reaction, a multi-component reaction between aldehyde, amine, and the ketone. As you can see here, the formation, there will be first the formation of in situ formation of the amine, and then the nucleophilic attack of the ketone of the enamine to the preformed amine. This will lead this very important building block for the synthesis of biologically active compounds. The success of this reaction is due to kinetic factors, as you can see here, that cross-Manic reaction is, the rate of cross-Manic reaction is higher than cross-aldol reaction. And also the mechanisms of all these reactions have been analyzed in details, as you can see, have been reported, the transition state, but we can skip and we can move on, because all these materials, you can find these materials in the book reported from Anastas. Let's move to another very important carbon-carbon bond forming reactions, in particular organocatalytic Michael reactions. This is one of the first examples reported in literature, also by very famous name, Professor Enders. And as you can see here, also in this case, it's a reaction between a ketone and a nitrostyrene. This is the Michael acceptor. In this case, the reaction is catalyzed by proline. And in particular, through the formation of the enamine, this is the transition state, it's possible to obtain the Michael adduct from moderate to good yields, good dastroselectivity, and only moderate enantioselectivity. I have to say that these reactions have been studied with several different organocatalysts, and the problem in organocatalysis is always to find the right catalyst for the right combinations of reagents. So this is the challenging part of the field. As in this case, do you hear me? Okay. As in this case, this is also an enamine catalysis in Michael reactions. You can see here nitrostyrene is a Michael acceptor, and the pronucleophile is aldehyde in this case. So passing from ketone to aldehyde, this is reported also by Hayashi, the catalyst, modifications of the catalyst will lead to better results, as you can see in this case. And in particular, the product, the Michael adduct, is obtained in very good yields, very good dastro-selectivity to very high dastro-selectivity, and outstanding enantio-selectivity. Okay. Now, let's move to another activation mode, and in particular to aminium ion catalysis, which is part of the covalent catalysis. And I'm going to show you some results reported by Barba's group in 2008. In particular, a Michael reaction between cinnamaldehyde, this is a Michael acceptor, and this is the pronucleophile. This is a particular compound, actually, it's a thioester. And now, later, a little bit later, we'll see why they use thioester in this reaction. In this case, the best catalyst is Jorgensen-Hayashi catalyst. It's always a prolinol derivatives. The TMS is trimethylcellulose, is a protecting group. As you can see here in this reaction, they obtain good yields, moderate diastro-selectivity. In this type of catalysis, aminium ion catalysis, diastro-selectivity is never so high, but usually enantiomeric excess are usually very, very high, as you can see here. And this is my little contribution to this field. Almost in the same time, we studied this reaction, this Michael reaction, asymmetric Michael reaction, with this particular pronucleophile is an immunoester, as you can see here. We obtained the product in good yields, usually in good yields, and these in very high yields. As always, the astro-selectivity, because we obtain two stereocenters in this reaction, the astro-selectivity is always moderate. Let me go a little bit in details, the mechanism. So this is a stepwise mechanism. I like to present the mechanism in this way. The first part of the mechanism is covalent bond formation between electrophile and the prolinol, as you can see here. Benzoic acid is a co-catalyst in this reaction, and under this condition, the product is aminium ion. Aminium ion is very reactive, is more reactive than a cinnamaldehyde. Water is also important, as you can see after. And the second part of the mechanism, starting from the pronucleophile, is acid-base reaction. So the pronucleophile should be enough acidic. The pKa of the pronucleophile should be quite low, because the base present in the medium are the secondary amine or benzoate, so they are not so strong bases. And this is an equilibrium with the formation of the carboanion, which is the nucleophilic species, and finally the pronucleophile. Do you hear me? Okay. The pronucleophile will attack the aminium ion from the lower side, from SI phase, because the RE phase is hindered by this very hindered moiety. So the attack is favored from the lower side, and for this reason, usually enantioselectivity is very high. The product of this reaction is this intermediate, and water is very important because it will give hydrolysis of this intermediate and favor the turnover. Okay, as you can figure out, pKa is very important in this field, and so all those compounds with the pronucleophile with the pKa lower than 17 can be used in this type of reactions. For example, simple esters like this are not reactive under these conditions. However, if you pass from a simple phenylacetic acetate to an acetate with the substitution of the aromatic ring with very strong electro-withdrawing group. These very strong electro-withdrawing groups will stabilize the carboanion, so the pKa of this compound will go a little bit down under 17, and so these compounds can be now used under these conditions, giving high efficiency, as you can see, high yields, always moderate diastro selectivity, but very high and
Hello, Antonio. Please unmute your mic. We can't hear you. Please unmute your mic.
Okay, do you hear me?
Yeah, now we can hear.
Because someone did it. I'm not done. Anyway, no problem. Maybe you have missed a little bit only this part, because this is just repetition. What I've said before is in amino ion catalysis, pKa or pronucleophile is very important. In the case of simple, if you want to use under this condition, these reactions, aldehydes or ketones, you can use a combination with enamine catalysis. Now, let's move a little bit further and let's consider different types of activation modes, and in particular in non-covalent catalysis. Non-covalent catalysis is characterized by ionic interactions, hydrogen bond formation, pi-pi staking, and so on. So, a combination of weak bonds, actually. So the knowledge about non-covalent catalysis is less precise than, of course, than covalent catalysis. Let me share with you a little bit of history in this field. This is one of the first papers appeared in the field, considering the use also of chinchon alkaloids. You can see here in the 80s. This is also a conjugated Thea-Michael reaction, and the results are only moderate. But what is important, they tended to rationalize the interactions, the mechanism in this reaction, proposing this transition state. In particular, you can see here chinchon alkaloids and the pronucleophile. So you have here a tertiary amine group here. This is a base. If the pronucleophile is sufficiently acidic, there will be acid-base reaction, leading this to ammonium salt. So the carboion anion is quite considered to be surrounded by a chiral pocket, and so it became a chiral itself. All the organocatalysis resemble a little bit the enzyme catalysis. Sometimes they share some mechanistic in common. In this, of course, the number of papers in this field is very huge. Let me show you also, some interesting results for me, and in particular this direct nitroaldehyde reaction, called also with the name Henry reaction. Is the reaction between nitromethane and, in this case, ketone, is cross-aldehyde reaction. And also in this case, as you can see, yields are very good, and as well as an antioselectivity. But the most popular organocatalysts in non-covalent catalysis are derivatives called bifunctional organocatalysts, known with the name of amine thiourea or urea catalysts. Some of them are reported in this slide, and most of them, as you can see, are commercially available. Some of them are very cheap, actually, so they are very popular. And in particular, this one is Takemoto catalyst, is cyclohexyldiamine derivative, as you can see here. In these catalysts, you will find always a basic site and an acidic site. This thiourea, you can note, is a hydrogen bond donor, so this will give very strong two hydrogen bond interactions with the electrophile. On the other hand, the amine group will activate the pronucleophile. This is the general transition state, usually accepted for this type of organocatalyzed reactions. And this is an example reported by Dixon, Colon, and Sauce. These are the most important name in this type of catalyst, organocatalysis. Also, Sauce is also a friend of mine. Is from Hungary, from Budapest. Anyway, as you can see, this is a Michael reaction between active methylene compound, as you can see, beta ketoester in enol form will attack this position is a conjugated addition. And the Michael acceptor is double activated by two hydrogen bonds, as you can see by thiourea group. Another very important way to perform organocatalysis is to use chiral phase transfer catalysis. This is actually very, very convenient. Let me show you some examples. These are the general mechanism. We can talk about phase transfer catalysis when we have two phases not miscible as an organic phase and aqueous phase. Some reactions are involved between a reagent soluble in organic phase and another reactant soluble only in aqueous phase. If this reaction is actually in an anion, the ammonium salt will favor the solubility of this anion in organic phase. Actually, there are several ways to describe this type of catalysis, and also this one with an interfacial region. And also this phase transfer catalysis is very popular when one of the phases is a solid, is insoluble solid like a salt. In this case, the chiral phase transfer catalyst will extract the anion from the solid, and so will favor the solubility of the anion in the organic phase. As you can imagine, this type of phase transfer catalysts, they are ammonium salts, as you can see, organic ammonium salts, usually derived from cinchona alkalides, as you can see here. These are very popular from the 90s, and you can see for sure some important names like Cori, Nobel Prize some years ago, and also this one reported by Maruoka. These are very cheap actually. And this one has been developed mainly by my friend Mario Vasa from the University of Linz, and also by myself. Also phosphonymous salts have been used in this type of chemistry. Let me show you a review in this field reported by Maruoka a few years ago. As you can see here, this type of catalysis is particularly useful in many different types of reactions like alkylation, conjugated addition, and so on, Manic, aldol, Dardens, and very big number of reactions. And some of them are really unique. And this is an example, is very, very important example, because this is an alkylation of glycine shift bases by benzyl bromide. You can use actually other bromide, other compounds in a similar way under phase transfer condition, usually toluene and water. As you can see, EE and antioselectivity usually very, very high. After the hydrolysis of this product, you will obtain the amino acid derivative. So this is one of the most important way for the asymmetric synthesis of amino acids. And you can synthesize amino acids from D and L series, and also several other unnatural amino acids that can be used in different types of pharmaceuticals and so on. And of course, when we are talking about pharmaceutical compounds, you know that one of the enantiomer is often more active than the others. And actually, presently, most of the drugs that appeared here, they are commercialized, they are in the market, they are color compounds. So asymmetric synthesis is very important. And it is very important to perform a asymmetric synthesis in a sustainable way. And also, some other reactions have been reported in this field, as you can see here. This is also reactions of glycine shift bases to obtain different types of amino acids, actually. I have finished this first part of the presentation, the general part of presentation, and my reference book is this one. This is green catalysis, and in a chapter of green catalysis, homogeneous catalysis, one of the most important chapters is the organocatalysis. And I also want to mention this book from Mario Vasa from the University of Linz about the applications of organocatalysis in the synthesis of natural products. And also there are several other books in asymmetric organocatalysis, as you can see here. Now, if I have time, let me show you some results from my recent research activity in this field. Let's start again with this reaction in the covalent organocatalysis. We have seen this reaction before between a microreaction between a cinnamaldehyde and this amino ester catalyzed by Jorgensen catalyst. After that, we studied different type of reaction. In particular, we studied we investigated the use of this type of heterocyclic compound activated with an ester group in several asymmetric reactions. Actually, this scaffold is very important heterocyclic compound known with the name of isondolinols and is part of several biologically active and pharmacological active compounds. We studied several reactions, but let's focus on microreaction and cross-aldol reactions. In particular, as you can see here in this slide, we studied microreactions between this pronucleophile and chalcone, is unsaturated ketones, alpha-beta unsaturated ketones. In these investigations, we investigated several types of organocatalysts because you don't know what is the best catalyst. You have to try several of them. You have only some genetically hints. You can move on which type of catalyst you can use. And actually, the best catalyst in this reaction was this one. Is always also derivatives from chinchone alkaloids, but in this case, the hydrogen bond donors are squaramide, are derived from squaramide, as you can see here. You can see here the scope of the reaction, the enantioselectivity, the adastroselectivity was always very high in this reaction, because in this reaction, we formed two new stereocenters. And the enantioselectivity was also good, usually around 80%, a little bit ease. We also were able to characterize the product by X-ray crystallography. X-ray crystallography is always very important, especially when you have to synthesize new product, you have to characterize the product. And we also used this type of pronucleophile in cross-aldol reaction, and in particular in cross-aldol reaction between with two benzaldehyde substituted in two positions with cyano group and carboxylate group. In this case, this is a one-pot reaction, is domino reaction, because we first obtain the aldol, but we do not isolate the aldol product The alkyl product with the give cyclization with the ester group, leading to this hybrid compound, a lactone. Also, in this case, we first obtain this compound, and then after hydrolysis, we obtain the same product. So it's a way to obtain this hybrid compound in two different ways. Of course, we studied these reactions from a symmetrical point of view. We used several types of organocatalysts, and the better results were obtained in the presence of 2-cyanobenzaldehyde. So we had to isolate this intermediate, and then after hydrolysis, we obtained this compound, which is pharmacologically interesting. The best results were obtained with the Takemoto catalysts, with the cyclohexane diamine compound derivative. And as you can see here, we obtained very high enantioselectivity, and also very high diastero-selectivity. It's not given in this slide, but in the next slide. Usually only one diasteromer was obtained, and in this case, as you can note, enantioselectivity is usually very, very high, most higher than 90%. And we wanted, again, investigate the reactivity of pronucleophiles in molecular reactions, in this case with cinemaldehyde. And changing the substrate, we had to completely change the activation mood, the organocatalyst. And in particular, the best catalyst in this reaction was this prolinol protected as TMS group, trimethylsilyl group. This is the classical Hayashi-Jorgensen catalyst. Under the optimized conditions, we did not isolate the Michael adduct, but we observed a further reaction, in situ reaction, between the aldehyde group here of the intermediate and the amide lactamine group, leading to this very interesting hemiaminal functionality in a tricyclic heterocyclic compound. Also, this case can be an interesting product for a further study of biological activities. We also studied, we had to, how can I say, characterize the product by X-ray, because at the beginning we were not sure by simple NMR what the product was. And as you can see here, in this compound, 3 new stereocenters are formed. So from a stereochemical point of view, the mixture could be very complex, but actually we observed the formation of only 2 diastereomers in the reaction media, and both of them usually in very high enantiopurity. So very interesting reaction also. And this is the mechanism, as you can see here. This mechanism is a little bit more complex of those one we have seen previously, but the general mechanism is based on the condensation between the cinemaldehyde that you can see here and the prolinol, okay, with the formation of aminium ion here. Then the carbon anion will attack the aminium ion from the lower side, leading to this first adduct in high enantioselectivity. And then after hydrolysis of this intermediate, because of water, you will obtain first this intermediate, and then this will give cyclization to obtain the final compound, which is very stable. We do not see aldehyde in this reaction, but only the hemiaminal functionality. Okay, let me go a little bit further. Let me show you another very important part, very important family of sustainable reactions. They are called one-pot reaction. Probably you know them. They are in this family. There are very subtypes of reactions known with the name of multi-component reaction, cascade, domino, or tandem reactions. All these reactions belong to this family, one-pot reaction. These reactions are particularly useful for the construction of very complex molecules in a single pot reaction, and often they are particularly useful in the construction of cyclic or heterocyclic compounds in a strategy of addition cyclization. What are the advantages from of these processes? In practice, you are able to construct a very complex molecule in a single pot. So there is no necessity, no need of the isolation of the intermediates. And so you will save time, first of all, but also solvents, reagents, and so on. So can be very, this is for me, these reactions for me are very nice reaction, and for me, it's very interesting research field. Up to now, they can be applied to a wide range of substrates, and they can consider, of course, sustainable processes because of atom, step, and pot economy, and because they avoid the purification of intermediates. And so, for this reason, they are particularly convenient for the synthesis of complex molecular architectures. Let me show you an example in which VJ gave very important contribution. You can see almost 10 years ago, very long time ago. Anyway, this is very nice reaction. Sorry, I need a little bit of water. Is a reaction between two cyanobenzaldehyde is a bifunctional electrophile and pronucleophiles. In this reaction, we have used actually several types of nucleophiles. So malonate, beta-ketoesters, diketones, also glycine shift bases, and also heteronucleophiles like primary amines. So in the case of primary amines, it is possible to obtain in a reaction with two cyanobenzaldehyde a reaction here, a compound here branched with the naming group. So it's aminal with an aminal group. What is the mechanism of this reaction? In this mechanism, of course, more than one functional groups are involved at the same time. The first reaction is acidic base reaction between the pronucleophile and the base. The base is usually potassium carbonate. So this is a very convenient reaction because you can use a tertiary amine like a triethylamine or potassium carbonate. So under very nice condition, the solvent can be used acetonitrile, or in some cases it's possible to use solvent-free conditions. So it's possible to perform this reaction without solvent. The first reaction is formation of carboanion here. After the protonation, this carboanion will attack the aldehyde group. This is aldehyde intermediate. We do not isolate this compound, but this will give cyclization at the cyano group with the formation of this imidate intermediate. Sometimes we are able to isolate the imidate, but however, if there is another proton, this is an acidic proton on the chain here, because of the base, there is an acidic base reaction with the protonation here and the ring opening, formation of this amide, and then this intermediate will rearrange in intramolecular azamycol reaction, leading in all the cases the isoindolinones in very high yields. Of course, we started to develop asymmetric versions of this reaction, but this reaction, the catalyst for this reaction should be again invented, because the transition state important for the formation of the new chiral center is the last one. So it is this intramolecular azamycol reaction. So it's very challenging. We have tried in this reaction, I think hundreds of catalysts. The best one is not commercially available, but it's possible to easily synthesize. This is an ammonium salt. So the reaction works well under phase transfer conditions in the presence of potassium carbonate, dichloromethane, and this catalyst. We were able to scale up the reaction to 2 millimole scale, so good scale. The product was obtained actually in quantitative yields and good enantioselectivity. However, what is important in this reaction is that after crystallization, simple crystallization process, it is possible to improve the enantiopurity of this compound up to 95% yield, very good enantiopurity, and still a good overall yield, 77%. This compound was particularly useful in the total synthesis, in asymmetric total synthesis of bioactive compound. Let me show you one example. Sorry. After the carboxylation here and esterification, this ester was selectively reduced with lithium boron hydride, leading to this alcohol. And after methylation and the displacement of this piperazine, is possible to obtain this compound, this very important compound, active on the central nervous system with unchanged enantiopurity and in overall yield of 50%. So this is the first asymmetric total synthesis of this type of compounds. These compounds have been studied more than 20 years ago. They were synthesized as racemate. And then the most active, the S enantiomer, was obtained after resolution of the racemic mixture in very low yield. So around, only the resolution was around 10% yield. So our pathway is very, very convenient. Now, finally, let me show you another very recent example. We moved to study the chemistry of another very challenging heterocyclic compound, and in particular, isoxazolone. These heterocyclic compounds are characterized, as you can see here, to exist in three tautomeric forms. So it's very difficult to handle this type of compounds with a proton present on the carbon number 4. If a proton is present here, you always have a mixture of tautomers in solution. So very difficult to study. But we wanted to study asymmetric Michael reaction of this alkylidene isoxazolone. So in the presence of an organocatalyst, the quinidine, this is the Michael adduct. And because there is a proton in this position, of course, we in solution observed the presence of three tautomeric form. But our idea was to entrap, was to use entrapping reagents at the end of the reaction that we checked by TLC. After the disappearance of the starting materials, we simply added one of these entrapping compounds. As you can see, there is a mistake here. Sorry, this is Bock. I done a mistake. Okay, I apologize. Anyway, methyl iodide and acetic anhydride. Sorry for the mistake. They will give selective entrapping of the enamine form of the nitrogen. So we were able to obtain this type of compound, which was characterized by X-ray, as you can see here, in quantitative yields, very convenient reaction, and good enantiometric excesses. But also in this case, what is important is that we were able to improve the enantiometric purity by crystallization, and after a single step of crystallization, we obtain almost this compound in a very high, only one enantiomer, with an enantiopurity higher than 99.9%. Very, very interesting. And this is the scope. You can see at the Bock protecting group on the enamine form of this type of compounds. And as you can see, we obtained from good to moderate enantiopurity, but in most of the cases, we were able to improve enantiopurity by crystallization. So this is my last slide. I could show much more results, but I think the time is going very fast. But let me give you some information about my group. This is a picture of my group before lockdown, and now it's a little bit changed because we had very, very strong lockdown in between February and at the end of April, everything was closed. Also, university was closed. But unfortunately, we managed in May to start again our activities. And up now, we have not stopped any activities, and we hope that we can continue. And up to now, of course, we will put masks in a photo like that. And I have one more PhD student and one more postdoc in the group. But I don't have the photo actually. And I have several, as you have seen, I have several collaborations with several other groups, especially in Europe and in Italy. Most importantly, with Mario Vasa from the University of Linz, and also some of them are missed in this slide. But for me are also important collaborations with the companies, and some of them are very, very famous, but they are established also in Italy, like Merck Seronos, and I can get some funds for my research. Let me advertise an international PhD because BJ was in Italy, because our BJ Moore was in Italy as PhD student, because he applied for an international PhD. Now, the next application, unfortunately, is in August next year. What is different, because this program a little bit improved with respect to BJ application, because apart from the salary, now there is a free accommodation, everything inclusive in our residence, in our accommodation, university accommodation. There is always a free meal per day in a canteen, and of course the possibility to travel not only in Italy but also in Europe with an improved salary. So this is actually very convenient, and if you are interested in organocatalysis and asymmetric synthesis as a sustainable processes, you can apply, you can let me know and apply after your master degree. And where is Salerno? This is, you can recognize Italy. This is surrounded by the sea here. The Salerno is the central south part of Italy on the coast, as you can see here, close to Naples. Naples is the capital of the area, and this is Salerno on the coast. Probably Vijay reminds this picture. And there are also some, from historical point of view, you can see there are possibility of sightseeing around the area. This is very ancient Roman town known with the name of Pompeii. As you can see here, these are not, they are statues, and these are paintings collected from the old houses. They are dated to more than 2,000 years. And also very close to Salerno, this is Greek temple in the south. Always, also in this case, was built more than 2,000 years ago. And also possibility to go to the beach, of course, during the holiday time, during the summer, and we have also very nice beaches on the coast. This year, of course, was a little bit more difficult to go to the beach, but fortunately we were able to manage some holidays with masks and with, how can I say, to keep a distance between. But fortunately, at the moment, the situation is not so bad in Italy. I have to admit it, fortunately. And this is the, you can see the pictures of our campus. Actually, it's a very modern campus with buildings, squares, and fountains. Also, the pictures are have been before lockdown. And also swimming pool in the campus. This is a library, it's not a factory, it's a library. This is the inside part of the library. And you can see here the labs with NMRs and so on, with pictures before, of course, lockdown. And thank you for your kind of attention. Where is the, I missed the, hello?
Hello, we can hear you.
Hello.
No problem.
But I don't
Nagaroj,
see,
sir.
okay. Okay.
Hello.
Do you see me?
Yeah, we can see you.
Yes, yes, sir. We can see
I've
you.
closed the presentation, and I want to thank in person. Thank you for your kind attention and for your kind invitation. If you have some questions, I can share again the presentation, and I hope to give you right answers. Okay.
Okay, sir. Thank you for the nice presentation. The session is open for the discussion. If anyone have the questions, then they can ask in the comment section, so that we can discuss the question with Professor Masa.
I hope everything was clear. And also, if some students, if there are students in line, if they have some curiosity, I can try to answer.
Okay, if there is no question, then we can move further. So thank you, Professor Antonio Massa, for the very nice, informative, well-organized, well-presented lecture given by you. It was a very nice research journey with you, with your presentation, with your papers, with your research lab. And finally, the pictures you have shown here of Italy, that was very nice. And I hope you are healthy, wealthy there, and take care of your health. Here in India also, now the situation is really difficult, but we can hope best for the future. So thank you once again, Professor Antonio.
Thank you very much. Good luck.
We'll move further the second session of this international webinar, and we have another eminent research person here, Dr. Bapurao Shinte sir from Department of Chemistry, Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad. So first of all, I will request Dr. A.B. Patil sir to introduce our second resource person for today's webinar. Dr. A.B. Patil sir, I request you to introduce Dr. Bapurao Shinte sir.
Okay, thank you, Professor Nagargoje. Am I audible to all?
Yes, sir.
Am I audible?
Yes, sir.
Yes.
Okay, good
Yes, sir, you are audible.
Good afternoon to all, respected dignitaries and all the delegates. I am Ashok Patil from KMC College, Khopoli. First of all, I would like to appreciate your active participation in the today's webinar on innovative research in chemical science for sustainable development. After excellent presentation by Professor Antonio Massa, now it's my privilege to introduce today's another eminent personality, Dr. Bapurao B. Shinte. Dr. Bapurao B. Shinte, presently working as Associate Professor at the Department of Chemistry, Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad. Dr. Shinte sir has pursued his PhD degree from National Chemical Laboratory and Savitribai Phule, Pune University, Pune. In addition to teaching to the postgraduate level, Dr. Shinte sir is having more than 18 years of research experience in the area of synthesis of bioactive compounds and new synthetic methodologies. During these teaching and research activities, sir has received various awards, and some of these are Shikshak Pratibha Puraskar in 2012, Indo-US Research Fellowship Award in the year 2013, Ideal Teacher Award in the year 2014, Research Professor Award in the year 2017, and Best Teacher Award in the year 2018. Now, in order to explore research and to ignite research attitude in the young researchers, Dr. Shinte sir has delivered more than 65 invited talks in various national and international conferences. At the same time, he functioned as resource person in more than 20 refresher and orientation courses. Dr. Shingte sir has not only chaired the sessions of various conferences, but also organized more than 7 conferences as well. Dr. Shingte sir is also functioning as a member of Board of Studies in Chemistry at different institutions in Maharashtra. He is a deputy coordinator of University Grant Commission Special Assistance Program for Departmental Research Support. Under his guidance, 5 students have been successfully awarded PhD degree, and various students are presently doing their research under the guidance of Professor Dr. Shingte sir. He is an editor of various international research journals like International Journal of Research Journal of Pure and Applied Chemistry. Dr. Shingte sir is reviver of more than 40 international journals, including Nature, Scientific Reports, and so on. Now, till today, Dr. Shingte sir has published more than 110 international research papers, including his research, recent international publication in the Chemical Reviews, with the total citations equal to 2,500, H index is 29, and I-10 index is 73. Now, with this brief introduction, I request Dr. Bapurao B. Shingte sir to deliver his invited talk, and his topic for today's conference is Rhodanine Scaffold in Drug Discovery. Thank you. Thank you, sir.
Thank you, Patil sir. I request Dr. Shingte sir to deliver their lecture.
Just 1 minute.
Okay, sir. Okay, sir.
Just I'm sharing my presentation to you. Yeah. Do you visible to my slides?
Yes, sir. Screen is visible.
Yeah.
Slides are not visible. We can see
Screen is
your
visible,
computer.
sir.
The screen is visible.
Just 1 minute again.
Now we can see your presentation, sir.
Yeah, thank you. Thank you. Okay. Good afternoon to one and all. Thank you very much, Patil sir, for nice introduction. First of all, I would like to congratulate both the colleges of different universities One belongs to Mumbai University and another belongs to SRTMU University Nanded, for a joint venture for organizing international webinar on innovative research in chemical science for sustainable development. That's why I am congratulated to Khopoli College as well as Devani College. We have listened about the managements from both the colleges, principals of both the college, heads of the departments, IQAC coordinators, and organizers from both the colleges. The inaugurator for this particular international webinar, Dr. Wagmare sir, Dean Faculty of Science and Technology from SRTMU University Nanded. The chairperson, respected Professor Bhaskar Davne sir, Director, School of Chemical Sciences, SRTMU Nanded. The organizers, my friends, Dr. Panjagale sir, Professor Amol Nagargoje sir, Dr. More sir, Dr. Patil sir, and all their colleagues from both the colleges. Thank you very much for inviting me for such a wonderful gathering of international peoples. And just we have listened excellent lecture delivered by the Professor Massa from Italy on asymmetric catalysis, particularly how to utilize chiral ureas for various reactions. Thank you very much once again. Am I audible properly?
Yes, sir.
Yes, sir. आवाज येतोय. Yes, sir.
Yeah. Okay. Yeah, before going to talk on this particular topic, Rodan in drug development, I would like to highlight our research contributions in synthetic as well as medicinal chemistry, as I have started my research career from the India's national institution, premier institution like National Chemical Laboratory, Pune, where I have worked on steroid chemistry, particularly on the generation of C20 chiral center. And this is the achievement in my research career, early research career started with the chemical communications, and we have reached up to the chemical reviews having impact factor 54. And then after joining as a faculty at Dr. Babasaheb Ambedkar Marathwada University in the Department of Chemistry, we have been working on different synthetic methodologies for carbon-carbon bond formation, carbon-nitrogen bond formation reactions. Various synthetic methodologies we have developed for the synthesis of various biological compound, and received such kind of output over here. Later on, presently our research group is focusing on the development of new chemical entities and medicinal chemistry, particularly triazole chemistry is our main target, and we have received a recognition in triazole chemistry by publishing such kind of well-reputed journals. Around 25 research articles on the triazole chemistry we have published. Then we will talk about as we are facing the problem of coronavirus COVID-19 pandemic. Not only this one, but also before this one, there are many more diseases. Not only India, but also whole world have faced such kind of problems. Particularly in India, top 10 causes of death in India, just we can see the major disease, that is heart disease, chronic obstructive pulmonary disease, diarrhea, stroke, lower respiratory infections, tuberculosis. Many more patients from the India, they are facing tuberculosis. It may be primary tuberculosis or MDR tuberculosis or XDR tuberculosis, many more types of TBs. Then early growing babies like neonatal preterm birth of various kinds of babies, they are also having facing some problems, and due to those problems they are getting died. Then self-harm, that is also one of the major cause of death in India. Around 1 to 2%, more than that one cases we have seen in India through self-harm. Then major diseases, road injuries. These road injuries, every day we are seeing many more accidents, and through those accidents many more people are facing their deaths through these road injuries, as well as other neonatal conditions. Through these 10 major diseases or major causes of death in India, we have seen. As I have talked about the tuberculosis, which is still India is facing a lot of problems, and still it's difficult to cure completely this TB. As not only India, but also many more countries, they are facing the problem of tuberculosis. Similarly, African countries, then Indian subcontinents, they are having major contributions in terms of tuberculosis. See, tuberculosis is one of the top 10 causes of death worldwide. It's one of the top 10 causes of death worldwide. Around in 2015, 10.4 million people fell ill with TB and 1.8 million died from the disease, including 0.4 million among people with HIV. Over 95% of TB deaths occurs in low and middle income countries like India. 6 countries account for 60% of the total, with India leading the country, followed by Indonesia, China, Nigeria, Pakistan, and India. And South Africa. Means India's contribution in terms of tuberculosis is more as compared to the whole world, around 60% our contribution. And till date, there is not a single drug which can cure tuberculosis. Okay. Then what are the drugs were used to cure tuberculosis? The first drug in 1943 is para-amino-salicylic acid by Lehman in 1943, first successful drug. Later on, streptomycin, which has made a remarkable progress in curing tuberculosis. Then later on in 1946, thiacetazole, and then most important first line drug, isoniazid came in 1952 and made a drastic change in curing tuberculosis. Then later on, pyrazinamide and rifampicin has totally changed the strategy in chemotherapy, pyrazinamide. And later on in 1961, ethambutol, later on fluoroquinolones, such type of drugs, first line drugs, second line drug, or combination of first line as well as second line drugs were given to the patients to cure tuberculosis. But unfortunately, there are various types of tuberculosis, not a primary tuberculosis, but MDR, multi-drug resistance tuberculosis, XDR, TDR tuberculosis, total drug resistance, means not a single drug or many drugs that cannot work for curing that tuberculosis. See, here is the list of various drugs. First line drug, isoniazid, rifampicin, ethambutol, pyrazinamide, streptomycin. These are the second line, and these new drugs, newer second line drugs, ciprofloxacin, ofloxacin, levofloxacin, these are the new drugs in combination of either first line drug or second line drug that doctors are providing to the patients, those who are facing either TB or MDR TB or XDR TB or TDR. Okay, not only tuberculosis, but also there is another disease where India is facing a lot of problems. Before that one, these are the structures of representative drugs of isoniazid, pyrazinamide, ofloxacin, kanamycin, rifampicin, ethambutol, bedaquiline. These are the either first line or second line drugs. As I told that second disease, that is important disease which India, not only India, but low middle income countries, they are facing fungal infections. There are 2 types of fungal infections. First one is a superficial fungal infection, and we know very well such kind of diseases in various peoples we have seen, and that is called as superficial fungal infection, which occurs in ringworm, skin, and mucus of the membrane. See, that is the fungal infection. Then another type of fungal infection, that is a systematic fungal infection, which occurs through Candida albicans, and which is the opportunistic infections. And to cure these 2 types of fungal infection, there are several drugs are available in the market to cure such type of fungal infections. Just I am providing to you the most important drugs from the category of antibiotics. Amphotericin B, nystatin, griseofulvin. These are the widely used antifungal drugs, but have some drawbacks of such kinds of antibiotics. That's why azoles came in the pictures, and different types of azoles are also used to cure fungal infection. For example, ketoconazole, itraconazole, miconazole, and many more azole antifungals were given to the patient for curing the fungal infection. Then allyl amines also is also used to cure the fungal infection, as well as pyrimidine derivatives. Pyrimidine derivatives are also used. Flucytosine is also used. Okay. As the title or the theme of this particular international webinar is a sustainable development, and to achieve that sustainable development, we have to think about the principles of green chemistry given by this great person, Paul Anastas, and these are the 12 principles of green chemistry. At least we have to follow some principles of green chemistry in our synthetic organic chemistry. That's why those blue colored around seven principles we have followed in the synthesis of our various compounds, like prevention, atom economy, less hazardous chemical synthesis, designing safer chemicals, safer solvents, auxiliaries, reduced derivatives, as well as catalysis. Just we have heard excellent lecture delivered by the Professor Massa on catalysis. Okay. After seeing these principles of green chemistry, as my talk is on rhodanine scaffold in drug discovery and development. Rhodanine. This is a somewhat a different scaffold in heterocyclic chemistry. This is actually 6-membered heterocyclic moiety having 2 sulfur atoms, 1 nitrogen, 1 oxygen, and it is exactly similar to 2,4-thiazolidinone. And this 2,4-thiazolidinone based several drugs are available in the market to cure diabetes, proglitazone, like that type of drugs are pioglitazone type of molecules are available based on thiazolidinone. It is similar to thiazolidinone, only the change in only 1 oxygen atom. In place of oxygen there is a sulfur, and this particular compound heterocyclic moiety, this is a saturated heterocycle discovered in 1877 by Nancy, and this small molecule can be prepared via different ways. Starts from this ammonium thiocyanate and alpha haloacetic acid, or there are another method, carbon disulfide ammonia, followed by treatment with sodium salt of fluoroacetic acid and acid hydrolysis. We can make this type of rhodanine moiety, or if you want to make unsubstituted rhodanines, you can start with the synthesis with the different kinds of amines, then carbon disulfide, haloacetic acid. You will have a substituted this particular rhodanine, N-substituted rhodanine. After seeing these particular methods to make such kinds of rhodanine, even though the basic skeleton of rhodanine, the 3 to 4 compounds, not only 3 to 4 compounds, but basic skeleton of this rhodanine is commercially available, and one can use directly this particular rhodanine. Okay, why we are working on this particular rhodanine moiety? See the picture of rhodanine chemistry, rhodanine publications on rhodanine. In the year of 2004, the number of publications is around 100, but in the year of 2014, you can see more than 450 publications, and in the year of 2019, around 1,500 publications on rhodanine research. We see the significance of this rhodanine scaffold in synthetic organic as well as medicinal chemistry. Okay, see, as rhodanine, this is the skeleton of rhodanine, and this moiety is related to aryalidine. Aryalidine is there, aryalidine, rhodanine is there. This aryalidine rhodanine scaffold displays various kinds of biological activities like anti-cancer. Simple molecule is there, small molecule. Such type of molecules shows anti-cancer activity, anti-HIV, anti-inflammatory, anti-hepatitis, anti-diabetic, antibacterial, antifungal, as well as Alzheimer activity. Means see the pharmacological significance of this aryalidine rhodanine moiety in synthetic as well as medicinal chemistry. See, there is one drug available in the market, ipalarestate. See the skeleton of ipalarestate. This is the rhodanine, plain rhodanine, and this one is acetic acid. And this particular compound is prepared from the derivative of cinnamaldehyde. See, aldehyde along with this rhodanine acetic acid. This is the methyl cinnamaldehyde, and this particular rhodanine acetic acid is prepared long back, and this particular drug is used for non-competitive and reversible aldose reductase inhibitor used for the treatment of diabetes neuropathy. See the significance of this particular small molecule, and by changing this skeleton, particularly aromatic skeleton, one can make thousands of the molecules. Okay. See, just representative compounds I will show you over here. See, this is the rhodanine moiety. This is our heterocycle, and here aryl group is there, means the whole skeleton becomes aryl rhodanine. And having some substituents over N substituent is acetic acid, and this type of compound is acting as an antiviral agent, and which is used for hepatitis C virus. Similarly, this is also a rhodanine moiety. Here is also rhodanine, but that rhodanine is substituted by means of some sulfonamide moiety over is here. Okay, not only this one, but also there are many more arylidine rhodanine compounds are here, which displays different kinds of biological activities, that is the DNA replicase, murodeligase inhibitors. Here see rhodanine moiety is there, simple rhodanine moiety. Here also rhodanine moiety, which acts as the HI1-GP4 inhibitor. This also rhodanine moiety. Here is also rhodanine moiety. See the simple molecule. It may be derived from trimethoxybenzaldehyde and rhodanine. That rhodanine is a substituted rhodanine. Along with see this EYA2 inhibitor, which is also having this rhodanine skeleton. After seeing the biological significance of such type of molecules, this one is again list of some active rhodanine based compounds. See the Cypher data for the year 2012 on arylidine rhodanine. Cypher data, all rhodanine compounds, around 1 lakh rhodanine compounds before February 2012. Means it definitely till date the number may be around 2 lakh rhodanine based compounds people have synthesized. Out of 2 lakh, only 10,000 compounds were screened for their biological activity. Means there is a scope to how to make similar molecules and have some biological activity, and may have you may have some other publication. Then commercially available rhodanine, see the number is again too much, around 66,000, and out of those 66,000, only 3,000 compounds were screened for their biological activity. See the significance of this particular small molecule in synthetic as well as the medicinal chemistry. Okay, then again some library of compounds having arylidine rhodanine is there, arylidine rhodanine, which shows better anti-tubercular activity. This arylidine rhodanine, here also arylidine rhodanine derived from some amino acid, aldehyde, and that particular. See again here, in place of benzene ring, some heterocycle skeleton is there. Here also some heterocycle, same type of molecules are there, and they are showing better anti-tubercular activity. Okay, then just we have seen simple arylidine rhodanine. Here again rhodanine moiety is there along with some quinoline. Means definitely to make such type of compounds, these peoples may have used quinoline based aldehyde along with this rhodanine, and simple novel type of condensation, and made such type of molecules and screen for their anti-cancer activity, and see the publication in AGMC. Similarly, such type of quinoline based molecule from this molecule derived some rhodanine, arylidine rhodanine, and displays better antimicrobial activity. Along with this this chloroquinoline based rhodanine derivative displays anti-infective activity, and see the outcome of this type of work in AGMC. Okay, then question may arise how such type of arylidine rhodanines we are prepared. Means different methods for preparation of arylidine rhodanine conjugates via novel condensation, simple novel condensation reaction between aromatic aldehydes with your like active methylene compounds, or the compound having alpha hydrogen atom or acidic proton is there. See, this is the simple transformation, and this transformation can be achieved under by acidic condition as well as the basic condition. Start from pyridine, piperidine, ammonia state in toluene or acetic acid by using Navier condensation method. Simple method is here. Okay. As we are talking about the principles of green chemistry and the significance of ionic liquids in organic synthesis, as we know very well, there are various ionic liquids. Those are commercially available, and in the decade of last decade of 20th century or first decade of 21st century, people have used a lot these ionic liquids in organic transformation to achieve diversely functionalized biodynamic heterocyclic compounds and screen for their biological activity. And we know very well the utility of these ionic liquids. Just I am focusing on only 3 ionic liquids and their synthetic utility in organic chemistry. See, the first ionic liquid is derived from DBU plus acetic acid. This is the ionic liquid, and see the utility of these ionic liquids for phosphamical addition reaction, diaryl ethers preparation, or 3,4,5 trisubstituted oxazolones. In the synthesis of such type of useful molecules, people have used DBU based ionic liquid, DBUH acetic acid, DBU acetic acid ionic liquid. Then another ionic liquid derived from DBU and sulfuric acid. This is the ionic liquid and used for some useful organic transformation. These are some representative examples where these 2 ionic liquids were used for making some great molecules in organic chemistry. And see the publications by utilizing such type of ionic liquids. Then the third ionic liquid, which is derived from triethylamine derived ionic liquid, triethylamine and sulfuric acid, and used for many more organic transformation. Around 60 organic transformations so far is achieved with the help of this ionic liquid, and these are some representative publications. Okay, after looking the principles of green chemistry, significance of this rhodanine chemistry, or how rhodanine type of compounds are giving better biological activity, then what is our contribution in this particular field? As my topic is rhodanine scaffold in drug discovery and drug development. See, as we have talked about some 3 to 4 slides before, in rhodanine, see here, we have designed the molecule having rhodanine moiety, like aradylene rhodanine. Here, quinoline is there. Is quinoline based rhodanine moiety. Means quinoline backbone with drug-like properties, and just we have seen such type of rhodanine scaffold displays anti-tubercular activity. Not only rhodanine, but aryllidine rhodanine type of molecules displays better anti-tubercular activity, and accordingly we have designed this type of molecules and synthesized with the help of, we have taken the care of principles of green chemistry and the use of ionic liquids for making such type of quinoline based rhodanine derivatives. See, this quinoline aldehyde is prepared from corresponding acetaldehyde via Winsper-Hug formulation reaction, followed by reaction of this 2-chloro-3-formyl quinoline with rhodanine. This is the rhodanine acetic acid and rhodanine propionic acid. 3 rhodanine derivatives we have used through Navier condensation approach by using DBU based ionic liquids under solvent-free condition at 80 degrees Celsius for 30 minutes. There is a carbon-carbon bond formation reaction takes place over here. Means these 7 aldehydes on reaction with these 3 rhodanine derivatives, we will have a total 21 quinoline based rhodanine derivatives in the presence of this DBU based ionic liquids under solvent-free condition. And see the structures of all these quinoline based rhodanine derivatives having a variation on this quinoline ring, methyl, ethoxy, chloro, methyl group, as well as there is a N substitution rhodanine acetic acid, as well as rhodanine propionic acid. Means we have prepared diversely functionalized highly substituted quinoline based rhodanine acetic acid and rhodanine propionic acid derivatives. See, chemistry wise, it looks a very simple use of ionic liquids for making such type of molecules. But after synthesis of these molecules and having characterization for all the compounds through H1NMR, carbon 13, HRMS, then to screen these particular compounds for different strains of fungi as well as bacteria, along with some TB strains, and with the help of some collaboration, we have screened all these compounds for different types of biological activities. First of all, we will see their antimicrobial activity. We have screened against the some bacterial strains along with fungal strains, but none of the compound is active against bacteria as well as fungi. Only one compound is active, that is a 32H, which is having only rhodanine skeleton. Rhodanine skeleton, only one compound is active against the bacterial strain, and none of the compound is displaying better antifungal activity. Only one or two compound is showing equivalent antifungal activity, means our antimicrobial activity is having negative results. Then see the specificity of our synthesizer compound against the antitubercular strains like MTBS37RA and M. bovis BCG strain. Two different types of strains are there, dormant and active for both these strains. And see the MIC as well as IC50 for some of the compounds derived from rhodanine as well as rhodanine acetic acid and rhodanine propionic acid. See the SAR, structure activity relationship over here. The compounds possessing only rhodanine, they are 32 numbered. The compounds having rhodanine acetic acid, they are 33, and rhodanine propionic acid 34A. But this acetic acid and propionic acid based molecules, they are inactive against these two strains. However, the compounds bearing rhodanine moiety starts from 32A to 32I. Out of that, the compound 32G is the most active compound with MIC value 0.9 microgram per ml against the M. bovis BCG strain. Against the MTB S37RA strain, see the MIC 3.5, IC50 2.3, MIC 2.8, 1.9, and see after studying, see the selectivity against the TB strain, not against the bacterial strain. Further, we have done molecular docking study against the enzyme of ZMP1 and having a good interactions with different amino acids. And this is the most active compound, and this is the docking image for this particular 32G compound against the active site of ZMP1 enzyme. All the synthesizer compounds, again, we have screened for their toxicity, site toxicity against the strain HUVEC, these 5 to 6 cell lines of anti-cancer, and none of the compound is toxic against these cancer cell lines. In short, in conclusion, out of those 21 compounds, we found that these 4 compounds are most active compounds with lower MIC value against both the TB strains, and this work we have published in the European Journal of Medicinal Chemistry in the year of 2017. Okay, from this particular work we inspired, why not to have some small modification in the skeleton of this particular quinoline, not in this rhodanine. We can make a small change over here, and accordingly we have designed the molecules, synthesized, characterized, and screen these compounds for different biological activity. And we will see that small change over here. See over here, in place of quinoline, we have made a small change over here. Tetrazoloquinoline we have made over here. Tetrazoloquinoline, and this tetrazoloquinoline, along with this rhodanine moiety, 2 pharmacophoric groups in a single molecular motif can enhance the biological activity of the respective compounds. And this is the concept of molecular hybridization. With the help of this molecular hybridization concept, we have made such type of compounds. See, This particular tetrazoloquinoline aldehyde is prepared from 2-chloro-3-formylquinoline via nucleophilic substitution of sodium azide and cyclation takes place, and we'll have such type of aldehyde over here, tetrazoloquinoline aldehyde. And again, similarly, reaction of this aldehyde with 3 rhodanine moieties, rhodanic acetic acid, and rhodanine propionic acid via non-linear condensation approach by using DBO based ionic liquid, that is a DBOH HSO4 under solvent-free condition at 80 degrees Celsius for 30 minutes, and we can have a library of compounds over here. You see, we can have around 21 compounds over here. See the different structures of and the modification on the tetrazoloquinoline over here, methyl or methoxy group, as well as fluoro group is also here. Okay, then as we have seen in earlier case where 2-chlorocysteine is there, most of the compounds they are active against the TB. However, small modification in that quinoline ring, we have made tetrazoloquinoline. This 36A compounds is related to rhodanine, 37 is related to rhodanine acetic acid, and 38 is related to rhodanine propionic acid. Again, rhodanine acetic acid, rhodanine propionic acid, they are inactive against the TB strain. However, one compound from rhodanine group, only 36 C compound is active with the MIC value is 4.5 and 2.01 microgram per ml against the 2 strains of TB, MTB, S37RA and Bovis BCG. However, we have seen in earlier case none of the compound is active against the fungal strains. Here, some of the compounds are active against the fungal strain, and see the specificity of such type of compound. Furthermore, the most active compound 36C, we have docked against the active sites of ZMP1 enzyme and having good interactions with the proteins of those with amino acids, and these compounds, synthesized compounds, we have evaluated for their toxicity, cytotoxicity with the help of MTT assay, have different kinds of cancer cell lines, and none of the compound is toxic against these cell lines. In short, in conclusion, for this particular compound, 4 compounds are active against the TB. 1 compound is against the TB strain, only 1 compound. However, remaining 3 compounds are some leads with lower MIC value, 25 microgram per ml against one of the fungal strain. And this work is published in the Journal of Bioorganic and Medicinal Information Letter in 2016. So far, we have seen the modification in the this quinoline or quinoline as well as tetrazoloquinoline we have used. Then, while going through the literature, we found that even though there are more than 1 lakh compound in the literature, but the simple aldehydes or the non-linear condensation of simple aldehyde with rhodanine, or rhodanine acetic acid or rhodanine propionic acid is well known in synthetic point of view. However, those compounds were not screened for their biological activity. That's why we have taken the advantage of better finding the literature and literature survey. We have made a similar type of compounds, known compounds. These all are known compounds with the help of non-linear condensation approach on these aromatic aldehydes, commercially available aromatic aldehydes with rhodanine and rhodanine acetic acid only in presence of triethylamine derived ionic liquid, triethylamine HSO4 at 80 degree again solvent-free condition. We have made 2 different types of rhodanine-based derivatives, and all these derivatives are well known. Only synthetic methodologies are different, but if we have not screened these compounds for their biological activity, this work will not go in such kind of journal. That work will go in the less than 0.7 or 0.8 impact factor journal. However, we have made all these compounds, and these are the structures of all these compounds, characterized very well and screened for their biological activity like antitubercular activity against the 2 strains of MTB S37RA and M. bovis BCG. However, we have seen one of the compounds, see here, 0.9 IC50 value over here. Means out of those series of compounds, we found that some of the compounds are there, showing better antitubercular activity. Okay, so far we have seen some rhodanine chemistry. It is similar to thiazolinone, and still we are working on rhodanine chemistry. Lot of molecules we have synthesized in our laboratory and screened for their different kinds of biological activity, and we got some lead molecules which I am not showing over here. However, the another similar moiety similar to rhodanine, that is a thiazolinone dione or the 4-thiazolinone molecules. We are working on that 4-thiazolinone also, and after synthesizing those molecules, we have screened those molecules for biological activity, either antifungal or antitubercular activity. And while synthesizing such kind of molecules, we have taken care of principles of green chemistry also. See, these 4-thiazolinone derivatives, well-known chemical entity in synthetic as well as medicinal chemistry and pharmaceutical chemistry. This is the basic skeleton of 4-thiazolinone compound, and while going through the literature, you will see more than 1,000 reports on synthesizing such type of molecules, either by variation in the aromatic aldehydes or variation in the aromatic amines, and accordingly, and variation in the chemical methodologies or synthetic methodologies. By changing some catalyst, you can have respective molecules and you will get some publication, no doubt. But if your literature is strong, then you will get better publication, and accordingly you have to work. This is the simple molecule prepared by the cyclocondensation of aromatic aldehyde, anilines, and thioglycolic acid. In presence of some acid catalyst, you will get library of such type of compounds. Accordingly, we have made this type of compounds simply by cyclocondensation of aromatic amines, aromatic aldehydes, thioglycolic acid. Those are available in the postgraduate laboratory, and accordingly we have made a library of compounds, around 20 or 21 compounds here. See, with the help of ionic liquid, which we have utilized in earlier synthesis, that ionic liquid is derived from triethylamine and sulfuric acid at 80 degrees 30 minutes, and within 5 to 6 days you can make a library of compounds, and these compounds are well-known compounds. We have characterized very well through IR, NMR spectroscopy, and later on we have screened all these diversely functionalized, means most of the compounds are halo substituted. You will see most of the compounds are halo substituted. Those aldehydes which we have used, they are halo substituted, either fluoro, bromo, or fluoro substituted compounds are there. And these compounds we have screened for their antitubercular activity, and we got some exciting results. And some of the two compounds, 10C and 10H, they are showing good antitubercular activity with lower IC50 value. See, against the TB strain MTB S37 REST. Okay. See, these are the most active compounds: 2.2, IC50, 4.2, 6.8, 5.C. And see the output of this particular work in the journal like Bioorganic and Medicinal Chemistry later, 2016. Okay, with this work we got inspired a lot. Why not to make some different types of thiazolinone derivatives and may displace better better biological activity, and accordingly we have designed the molecules based on some skeletons. And see over here, this is the drug available in the market, that is a LL3858, the first drug. And see the skeleton. This skeleton is isoniazid, which is anti-tubercular drug widely used for curing TB. This is the INH based drug. Here also isonicotinic acid hydrazide, INH is here, and people have made hydrazone from INH and along with aldehyde. See over here, this INH based molecule, as well as over here, see, this is a isonicotinic acid hydrazide derived some hydrazones, and they are showing better activities against the different TB strains. Along with, see over here, simple the molecule derived from INH, para-hydroxybenzaldehyde, and that hydroxyl group is utilized for making this amide as well as ether linkage. Beautiful molecule, small molecule, people have derived. And from this literature we inspired why not to make some INH based molecules along with some other pharmacophoric group. See over here. Further report, again INH is there along with this thiazolinone moiety is there, thiazolinone. Along with INH and some different types of 6-membered heterocyclic moiety as well as 5-membered. And with this literature survey, why not to have this INH based thiazolone molecules? Accordingly, we have made the compounds, we have designed the compound, see, and the backbone just we have modified over here. Rather than taking simple aromatic aldehydes, we have taken tetrazoloquinonyl aldehyde. Then we have this INH skeleton. Not only INH, but we have also modified this INH. Is this carboxylic acid hydrazide group is at 4 position with respect to this pyridine nitrogen. We have varied the position and we have synthesized such type of molecules as we know the importance of thiazolone. INH as antitubercular drug. This tetrazoloquinone is a drug-like property this shows, and accordingly we have synthesized. See over here. This tetrazoloquinonyl aldehyde, as we have seen the synthesis of that one, along with this acid hydrazides, we have taken 3 acid hydrazides. One is isonicotinic acid hydrazide, another one is a nicotinic acid hydrazide for SAR purpose, as well as similar to INH, we have used a para-nitrobenzoic acid hydrazide along with this 5-glycolic acid in presence of DBU-based ionic liquid, that DBUH acetic acid at 80 degrees Celsius for 1 hour, and we have made a library of derivatives at under solvent-free condition with the help of some 6 aldehydes, 3 acid hydrazides, and 1 5-glycolic acid. And we have made different 18 derivatives. See, different 18 derivatives. This one is simple INH based. This one is A is INH based, B is nicotinic acid hydrazide based, and this C is para-nitrobenzoic acid hydrazide based. These are the structures, and after synthesizing such type of molecules, we have characterized for their H1 NMR carbon 13 as well as HRMS. Then, after synthesizing these library of compounds, we have screened these compounds for their anti-tubercular activity against the strains of TB, MTB, S37RA, and M. bovis disease strain. Along with positive control is the rifampicin, and we have made this kind of derivatives. And see, you will see something over here. 19A derived from INH, 19B derived from nicotinic acid hydrazide, and 19C derived from para-nitrobenzoic acid. See over here, the compound derived from INH showing lower IC50 value 0.36 than these two. Again, D compound INH based. This E compound and F compound, they are nicotinic acid hydrazide and para-nitro. Again, this 19G is derived from INH. Again, this 19J derived from INH and showing better antitubercular activity with lower IC50 value 0.15. Again, these two are inactive, then third one is 0.26. Again, these two are inactive, again, third one is most active. See, means the thiazolinones derived from INH as well as tetrazoloquinonyl aldehyde and thioglycolic acid, they are showing better antitubercular activity against MTBS37, and the same trend we can see in another strain of TB, that is M. bovis disease strain. This is strain that is 0.69, 0.07 C over here. Against this particular strain, the MIC value for this 19D compound is similar to standard drug, that is rifampicin, 0.07, 0.03 C over here, 0.03, 0.377, 0.06, 0.03 means these compounds are showing equivalent activity against the TB strain. Okay, all the synthesized compounds were docked against these enzyme or active sites of MTB INHA enzyme, and they are showing a better docking score against these particular enzymes. Okay, these synthesized compounds were screened for their toxicity, and none of the compound is toxic, and this is the lead compound we got from this thiazolone along with INH based skeleton C. We have used a total 6 different types of aldehydes, and we got 5 lead most active compounds against the two strains of TB, that is MTBS 37A and MBOVIS BCG strain, and this work we have published in the Journal of Medicine, Chemistry, Communication. Now it is changed RSC medicinal chemistry. Okay. Furthermore, again from this thiazolone chemistry, we inspired why not to have some another type of derivatives. And while going through the literature, in many more cases, we have seen such type of skeleton reaction of some aromatic amine, aldehyde, and thioglycolic acid. We got such type of literature over there. But here we have gone through specific literature. Here sulfonamide or sulfonin incorporated for thiazolone. See over here, sulfonamide group. Sulfonamide, here sulfonin group is there and thiazolone is there. Here also sulfonyl group, here also sulfonyl group, here also sulfonamide, and here thiazolone. And we found that only two compounds so far in the literature. Before our work, only these two compounds derived from this chlorofluorobenzaldehyde, then tosyl hydrazide and thioglycolic acid. This compound, as well as the dichlorobenzaldehyde, 2,6-dichlorobenzaldehyde, this tosyl hydrazide and thioglycolic acid. Only two compounds were known in the literature, and they are showing some activity against some strains. And from these two structures, we got inspired to make some similar type of library, and why not to have a screen these compounds for some biological activity. Accordingly, we have made a plan like sulfonamide, then thiazolone, and some aromatic compounds. We have designed such type of molecules and synthesized. See the aromatic aldehydes, this tosyl hydrazide, then thioglycolic acid in presence of already synthesized TBU-based ionic liquid, TBUH, HSO4. Cyclocondensation reaction at 80 degrees Celsius for 1 hour under solvent-free condition, and we have made such kind of library like 13 derivatives we have made, and well characterized. All these compounds are new. Only two compounds are known in the literature, that is dichloro and chlorofluoro-based compound, and whatever the compounds we have made, they are unknown. And see, the researchers can see there is a scope to make different types of sulfonyl hydrazide based thiazolones. See, just you can modify the aldehyde. Accordingly, we have made this simple type of library of compounds and characterized very well through NMR, C13, and mass, and screen for their antifungal activity, antioxidant activity, and we found that some of the derivatives, they are showing equivalent antifungal activity against the fungal strain like mycorrhizal, fluconazole, and some of the derivatives, they are showing better antioxidant activity against the strain of DHT standard drug, that is positive control butylated hydroxytoluene. Some of the compound like this 15D is showing equivalent activity for antioxidant, and some of the compounds, they are showing equivalent antifungal activity. And the active compounds, we have talked against the active site of sterol 14 demethylase cytochrome P51, and they are showing better binding interactions with the different types of amino acids. And see, these are the most active compounds, 6 active compounds. Then you can have some structure activity relationship, or you can make some QSR, and you can have a library of compounds where sulfonyl hydrazide based thiazolone known. So you can make a library of compounds, and this work we have published in the New Journal of Chemistry, which is published by RSC in the year of 2016. Okay, so far we have seen some rhodanine based derivatives, rhodanine acetic acid, rhodanine propanic acid, having quinoline skeleton, as well as tetrazoloquinoline skeleton, along with some thiazolone skeletons over here, thiazolone, as well as tetrazoloquinoline, thiazolone, along with INH, as well as nicotinic acid hydrazide and para-nitrobenzoic acid hydrazide we have seen. Along with we have seen this sulfonyl, means tosyl hydrazide derived new thiazolones, and synthesis we have seen their antitubercular activity, antioxidant, antimicrobial, including antifungal, antibacterial, along with cytotoxicity as well as molecular study we have seen for this type of library of compounds. Okay, Before going to talk about this conclusion part, similar type of library we have made in our laboratory, and thousands of compounds are available with us, and we are ready to collaborate the peoples from the biology section, particularly microbiology, biotechnology, biochemistry. We are ready to collaborate with them to carry out some biological activity like anti-cancer, antifungal, antimicrobial, anti-tuberculosis, anti-inflammatory. We are facing a lot of problems to have a collaboration with the experts from the India as well as abroad, and we are ready to collaborate these papers. Lastly, I would like to thank all my research students from our group, those who have contributed in the designing and development of such kind of library of compounds. And whatever work I have shown over here is carried out by one of my students, Dr. Jnaneshwar Subedar, and just his one chapter of his thesis I have presented over here on rhodanine and thiazolidinone, and thanks to Subedar and remaining peoples, along with my collaborators from different institutions, Dr. Dhiman Sarkar for anti-tubercular activity, Jayaprakash Sanseti for antifungal as well as docking study, Mukund Deshpande from NCL Pune for antifungal activity, Vijay Khadkar from Vishwakarma University Pune for molecular docking studies, Sandeep Shinde now is in France. We have worked with each other for some antifungal, antibacterial activity along with Prakash Jha. And now we have many more collaborators are there, but whatever collaborations are there through this work, just I have presented over here. And finally, I would like to thank to the organizers for giving me an opportunity in this particular international webinar on sustainable development and innovative ideas in chemical sciences. Thank you once again for the organizers, principals of both the colleges, then heads of both the colleges, then coordinators of both the colleges, along with you all audience and as well as faculty members from various affiliated colleges, university members, and my dear colleagues. Thank you one and all. Thank you.
Thank you, sir, for the very nice and informative lecture. As usual, you deliver everywhere. And now the session is open for the discussion. If anyone have some question, then he or she can ask the question so that we can discuss here.
I am ready to talk with the young budding peoples of India, researchers from the India.
If there is any doubt, then you can ask the question. The session is open for the discussion. I think no question is there, so we will move toward the last part of this international webinar. Thank you, sir, for the very nice lecture. Someone is having question, I think.
Yeah, take his question. Because I don't have chat box.
Panchagale, sir.
Okay.
Allow him to.
Who is that one?
Yes, Mr. Chandu Potangale.
Okay.
Hello.
Hello. Can you hear me, sir?
Yes, sir. Your voice is audible. You can ask the question.
Yeah, sir. What happens in the most of the academic research institute when you try about this scaffold derivatives and library synthesis? People synthesize the more compound, but the compounds are not screened more. Means they are not going to the final stage. Any drug is not coming out of all this scaffold synthesis or this library synthesis. I mean, lead compounds are not forming. Like NCL, they may have the facility, but if you go in the universities or research institute, academic institute, we are not going to the final stage.
Yeah. Your question is valid, sir. Just I want to answer to your question. This is the question not only of you, but this is the question of whole India. I can consider like that way.
Yeah.
We are doing academic research, which is useful to have some modifications in the structure, and industry peoples can take the advantage of this basic research. Industry peoples can take the advantage of this basic research, and accordingly they can modify some molecules, and in future they may get some leads. And that industry people see the drugs available in the market, they are very small, and before coming those drugs in the market, someone may have synthesized in the laboratory, in the university or some affiliated colleges long back. See, even though we are listening about the repurposing of drug for this COVID-19, so many drugs earlier days they have prepared for some different purpose, but they are inactive. But nowadays, these same drugs is given to cure the COVID-19. For example, remdesivir. Example I am giving to you, remdesivir. Even though people don't know about the remdesivir in the last 5 to 6 years, purpose of making that remdesivir for a dengue purpose. But the real utility came in for this coronavirus COVID-19. I'm coming back towards your question. Even though just we are providing some skeleton to the industry people, those who are pursuing advanced research, and from this skeleton, small molecule can get in future. That is the idea of making such type of library of molecules.
Okay, thank you, sir. Thank you very much, sir.
Thank you, sir.
If any question, then you can ask. Write in the comment section if you have any questions, so that you will be unmuted. Okay, should we move further? Okay. Thank you, sir, for the nice lecture, and good discussion is over at the end. So I request now to give vote of thanks to this international webinar finally. Our one of the...
First of all, you have to take the remarks from chairperson.
Yes, sir. Yes, sir.
If Daune sir is here, then you can take the feedback from chairperson.
Yes, sir. So I request Professor Dr. Daune sir to make some remarks here at the end of this international webinar.
I think Daune sir is not in the meeting now.
Okay. Let it be.
Okay. So I request finally our colleague, and who have taken millions of effort to make this webinar happen. So I request Dr. Sharad Panchagale sir to give vote of thanks and conclude the session.
Thank you, Nagaruji sir. Now we are at the conclusion of webinar. I am here to express the vote of thanks. On behalf of organizers, I take this opportunity to thank Professor Antonio Massa for giving wonderful lecture on principles and application of asymmetric organocatalysis. Thank you, sir. I'm also thank other resource person, Dr. Bapurao Shingte, for very nice lecture on rhodanine scaffold in drug discovery. Thank you, Bapurao Shingte, sir. I'm thankful to Professor Dr. L.M. Wagmare, sir, for inauguration of the webinar. I express my sincere thanks to Professor Bhaskar Daune for chairing the session. I'm grateful to Mr. Govindrao Bhopnikar, sir, Mr. Gajanan Bhopnikar, sir, of Janaseva Sevabhavi Pratishthan, Bhopni, and Mr. Santosh Jangam, sir, Mr. Kushor Patil, sir, Mr. Sanjay Patil, sir, Mr. Rajesh Abani, sir, of Khalapur Taluka Shikshan Prasarak Mandal Khopoli for encouragement and support in organizing this webinar. I thank Principal Dr. Chandrakant Javle, sir, and Principal Dr. Mahesh Khanvilkar, sir, for their guidance and financial support for organizing this webinar. I remain grateful to my friends, Dr. Vijay More, Mr. Ankush Bhusawale, Mr. Ankush Bhaskar, Dr. Chandrasekhar Malba of Kailaswasi Rasika Mahavidyalaya Devni, and Mr. Amol Nagaruje, Dr. Ashok Patil, Dr. Deepak Gaikwad of KMC College, Khopoli for their sincere efforts in organizing this webinar. Lastly, I thank all the participants, and with this, I announce this webinar is concluded. Thank you. I have one more announcement. The feedback forms will be sent through mail to all the participants. Thank you.
Bye bye.
Thank you, sir.
Bye.
बोला सर.
अभिनंदन सगळ्यांचं.
Thank you.
Thank you, sir. Thank you.
तर मोरे दिसत नाही. मोरे अभिनंदन दोघांचंही. Very nice conference you have organized and given an opportunity to listen the person from Italy. Okay, that's the opportunity. People of Maharashtra.